Hello students, in the last lecture we can study the topic that is the multiple alleles. Definition of multiple allele, its characteristics, and we can study the two examples of multiple allele. First one, blood groups in human beings and coat color in case of rabbits one. So in case of blood groups we can observe that is the four blood groups are observed and these blood groups are given by Carl and Steiner. And second one example that is the coat color in rabbits, we can observe the four coat colors whether it is the agouti or white, chinchilla, Himalayan and albino. So dominance which is observed in case of coat colors as according to the series which is given one. And then we can study the difference between qualitative and quantitative character. Some points we can study some points which are remaining in the last lecture. We have to study in this particular lecture in case of the difference one. So first one point in case of qualitative and quantitative character, whether this difference may be asked as a quantitative or qualitative, or whether it is asked as a qualitative or quantitative character one. First important point in case of difference, that is the qualitative characters are controlled by one or few genes. They are called as a oligogenes. And in case of quantitative, these characters can be controlled by many genes. So they are called as a polygenes one. So whether difference is also asked as oligogenic character or polygenic character because of the control of the one gene or many genes one. Second one important difference will be qualitative characters are easily seen with the help of the eyes and we can classify them into the distinct groups. It is possible one. But in case of quantitative character, it is difficult to classify the quantitative characters into the groups one. That is the easily seen means that is the we can easily observe the flower color or seed coat color or seed surface whether it is a rough or smooth one. But in case of the that quantitative character, it is difficult to that is the seen whether height or weight or yield one. We can assume that particular character or we can measure that character one. Third one, that is the this character possess the discontinuous variation. Discontinuous variation means there will be the no change in the flower color in case of seed surface one. But here in case of quantitative, it will possess the continuous variation. Yield may be variable one, height may be variable one. So continuous variation which is observed in case of quantitative character one. Fourth point will be we can measure the this qualitative character by units. It is not possible one. In case of centimeter or kg, we cannot possible to measure that particular characters one. Suppose flower color or seed surface one. But in case of quantitative character, we can measuring the that character with the help of the units, measuring unit like centimeter, kg or quintal one. Height of the plant we can measure in centimeter or meter one. Or yield we can measure in case of grams, kg or quintals one. Then environmental effect is not observed in case of qualitative character. That is the no effect of environment which is observed in case of qualitative character. But in case of quantitative character, environmental effect is observed one because of adverse environmental condition. Weight of seed or yield of the dead crop which is adversely affected one. And last point, that is the some of the examples in case of quantitative and qualitative characters are given. In case of qualitative character, flower color, seed coat color or seed surface. These are the some of the examples. And in case of height of plant, weight of pod per plant or yield, these are the examples of quantitative character. So always the difference is asked in case of the that qualitative characters or quantitative character one. So in that particular topic, that is the multiple allele, question will be always asked for descriptive, define multiple allele, write down the important characteristics of multiple alleles with examples. Then sometimes short note may be asked blood groups in human beings or difference may be asked, that is the qualitative characters and quantitative characters one. The next point we can study in case of the that particular genetics, that is a multiple factor hypothesis. So factor is nothing also it is also called as a gene one. So multiple gene hypothesis or multiple factor hypothesis, that is the concept which is given by the scientist Nielsen Ill, and on the basis of the results or results of that experiment he can give on this multiple factor hypothesis. So in this hypothesis he can assume that a given quantitative character which is controlled by series of independent genes which is having additive or cumulative effects one. And in supporting to that statement, he can give studied the two example, that is the kernel color in width and ear length in case of the mage one. So in this particularly lecture, we can study one example, that is the kernel color in width, and we can prove that is the series of independent genes are involved in case of the genotype. They will show the additive and cumulative effect. So statement of that hypothesis, that is this hypothesis assumes that given quantitative character, that is the which is measurable one, that quantitative character which is controlled by series of independent genes. Independent genes means their number may be variable one. So on the basis of their number which is present in that genotype, that shows the different effects one. So how these effects we have to observe by studying the one example in case of the kernel color in wheat. So in case of wheat, suppose we can consider capital R1 and capital R2. These two dominant genes are responsible for red color development. Then when we can make a cross between red kernel variety with white kernel variety, the F1 shows intermediate effect, that is the light red color. Because of the intermediate color effects we can observe, that is the light red color one. So this light red color which is obtained in F1 generation, and again when we have to done the shelping or for obtaining the F2 generation, when we can make a cross between F1 into F1, we can observe 1:4:6:4:1. Total individuals are obtained in F2 generation are 16, but ratio should be modified 1:4:6:4:1 because of the multiple factor or multiple gene involvement one. So how this multiple gene involvement which is occur, we can study one. Suppose parents will be dark red and white. First parent is female, second parent is male. Always in crossing, first parent is female and second one is the male one. So parents are dark red crosses with white. So when we can make a cross, that is the dark red into white. Suppose for homozygous genotype we can take R1 and capital R2. These are the two gene involvement. So homozygous genotype will be capital R1, capital R1, capital R2, capital R2. Homozygous genotype. Then second one parent is the white. So for white we can take the recessive genes, small r1, small r1, small r2, small r2. Both genotypes are homozygous. Two alleles are involved. Capital R1 is the first and second capital R2. So gametes will be capital R1, capital R2. And for a white, small r1, small r2. So in that particular slide there is the correction, that is the always gametes are written in circle. So capital R1 and capital R2 having a circle, and small r1, small r2 having a circle one. First is the female gamete, second one is the male gamete. When there is the union of gametes will be occur, the F1 generation which is obtained or new individual which is obtained, that is the capital R1, small r1, capital R2, small r2. This is the heterozygous genotype which is obtained in F1 generation, and it shows the intermediate effect, and that effect will be light red. So how this light red which is arises in F1, that will be observed in next one, that is the uh, color effects we have to observe in the F2 generation one. We can understand when we have to study the F2 generation. Then when we have to proceed for F2 generation, how we can obtain the F2 generation? By shelping of F1 or by crossing of F1 into F1. Already we can determine the F1 genotype, which is the F1 genotype capital R1, small r1, capital R2, small r2, which is crosses with again F1, same genotype capital R1, small r1, capital R2, small r2. But both the genotypes are heterozygous. So two heterozygous genes are present in that both genotypes. So because of that, that will produces the four gametes. First F1 produces four gametes, second F1 produces four gametes. So which are that four gametes? Capital R1, capital R2, this is the first gamete. Second capital R1, small r2, this is the second gamete. Third gamete will be capital R2, small r1, and fourth one is the small r2, small r2. So again second F1 will produce a same four gametes. So we can make a circle to the all that four into four gametes. So first F1 produces 4, second F1 produces 4. So 4 into 4, 16, genotypes are obtained in F2 generation with the help of the checkerboard method. So within the that 16 genotype, we can make a sorting one. So likewise in case of dihybrid cross, we can obtain the 16 genotypes with the help of the checkered board. And within the 16 genotypes, when we have to sort out that 16 genotypes, we can obtain the ratio, that is the dihybrid ratio, 9:2, 3:2, 3:1. So 9 plus 3 plus 3 plus 1 is equal to 16. Here also ratio, that is the 16 genotypes are obtained, but on the basis of the independent gene involvement, ratio should be modified as a 1:2, 4:2, 6:2, 4:1. Total individual may be 16 one. Suppose when we have to sort out that genotypes from the checkered board or from 16 genotypes, first genotype in F2 generation, we can observe on screen, that is the one which is the similar capital R1, capital R1, capital R2, capital R2. Its phenotype will be dark red and its genotype will be only 1. Ratio will be considered 1 and number of dominant gene will be 4. So in that genotype, number of dominant genes are 4. How it will be 4? Capital R1, capital R1, capital R2, capital R2. Here 4 dominant genes are present. So because of that 4 dominant genes, this genotype shows the color effect, that is the dark red. Second 4 genotypes we can observe, that is the 2 1, capital R1, capital R2, capital R2, small r2. And second two are capital R1, small r1, capital R2, capital R2. These 4 genotypes are red one, only red one, because there is the presence of the 3 dominant genes. Which are the 3 dominant genes? Capital R1, capital R2, capital R2, or whether capital R1, capital R2, capital R2. So 4 genotypes in which 3 dominant genes are present, so color effect is the red one. So here up to this ratio will be 1:4:1. Next genotypes we have to consider, that is the 4 genotypes which are heterozygous. Which are these? Capital R1, small r1, capital R2, small r2. And next genotype which is in one number, capital R1, capital R1, small r2, small r2. Again one genotype will be small r1, capital R2, capital R2. So within the that 6 genotypes we can observe the 2 dominant genes are present, so color is the light red. Likewise in F1 we can observe only presence of the 2 dominant gene. Which are that 2 dominant genes? In 4 genotypes we can observe capital R1, capital R2. Then in next one genotype, capital R1, capital R1, and again in one, capital R2, capital R2. So these 6 genotypes in which 2 dominant genes are present, so its color effect will be light red one. Then next 4 genotypes we can consider, that is the 2 1, capital R1, small r1, small r2, small r2, and again in 2, small r1, capital R2, small r2. So within the that one, only one dominant gene is present, and these genotypes are 4. So its color effect will be pale red because of the one dominant gene. And last one genotype, which is the recessive homozygous, which is observed in one number, small r1, small r1, small r2, small r2, that is the white one. Here number of dominant gene present will be 0, so its color effect will be 1 1. So here in the F2 generation we can observe the ratio 1:4:6:4:1. So from that it is clear that 4 dominant gene produces deep red or dark red color, 3 dominant genes produces red color, 2 dominant genes produces light red color, 1 dominant gene produces the pale red color. The recessive gene or absence of the recessive, that is the dominant gene, produces the no coloration, that is the white color. So from that clear it is the number of the indominant genes or independent genes, they have additive and cumulative effects. And when multiple factor or multiple gene involvement is there, which is given by Nielsen yield, we can observe a ratio of 1:4:6:4:1. So different color shades are observed in F2 generation, whether it is from dark red, then red, light red, pale red, and last one is the wide. So ratio should be modified 1 as to 4 as to 6 as to 4 as to 1 in F2 generation when multiple factor hypothesis operates 1. So we can solve that example by taking the genotypes, then find out the gametes. And when we have to find out the gametes, in checkered board, in horizontal column we can enter the female gamete, and sorry, in vertical we can enter the female gametes, and in horizontal column we can enter the male gametes, and just to do the multiplication indicates by crossing female parent with male parent and return the genotype in respective checkerboard. We can obtain the 16 genotypes, and within the 16 genotypes just we have to sort out likewise, which is given on the screen. We can obtain 1 as to 4 as to 6 as to 4 as to 1 ratio on the basis of the number of gene involvement. So this is the multiple factor hypothesis. Okay, thank you.